I'm not going into detail. We were just interested in some cardiovascular diseases, which is not the topic, not the mainstream of the laboratory, Professor Foster's laboratory, because she's the person who is doing a lot of stuff for the blood-brain barrier. And here we're just doing the research on Takotsubo syndrome. Takotsubo syndrome was actually discovered in Japan, and Japanese are working very hard. You already know that. So they have a lot of stress, and from the stress, they have a problem with the cardiovascular system. And this is one of the examples why they might also live shorter than normal life, of course. They have some kind of balloon heart, and myotrophy, cardiomyotrophy, so the muscle becomes weak, and you will have the patient might prone to stroke and some other different nested things. And we had in Germany also some subpopulation which are suffering from this particular disease. We got some groups of patients with the Takotsubo syndrome in Germany, and we were thinking, what's going to happen with these patients? What is the pathogenesis of this particular disease? Because right now, the hypothesis is the kind of pathogenesis for this particular disease is the kind of inflammation, immune inflammation. So a lot of immune factors, pro-inflammatory cytokines are releasing because you have adrenaline in the system, and you kind of cytokine storm, and this will give you the particular balloon heart. And we try to figure it out if we got what's happening with these patients. And so we created some kind of in vitro assay where we have different concentrations of catecholamines. This is the kind of, not physiological, this is the kind of a positive control. We just treating cells with the catecholamines and see what is happening with the blood-brain barrier biomarkers. These are all blood-brain barrier biomarkers because we are interested in blood-brain barrier, and these are tijunctional protein and some proteins which are expressed at the interface of the endothelial cells at the blood-brain barrier. And we have also positive control, kind of a negative control, postmenopausal women, and concentrations from the acute and chronic Takotsubo syndrome patient. And what we have here, we actually don't really see any effect, because people are claiming in the literature, people are claiming if you have a patient with the Takotsubo syndrome, usually you have a problem with the blood-brain barrier proteins. The blood-brain barrier becomes compromised, and you got the negative effect coming from different toxic substances going to the brain, because the blood-brain barrier is not protecting you anymore. And as you can see here, this is not the case. So what is the case? And the case problem might be that the person is very old. So we might think that this would be very important pathogenic factor. That when the person is getting older, then it could relate to the development of Takotsubo syndrome. This was kind of a finding, the main finding. And then we also try to implement bioinformatics, it's not chemoinformatics, it's pure bioinformatics and protein interaction, protein microRNA interaction network. I was doing it using some open source RNA like Mirna and some cytoscape profiles, because we had a gene expression profiles. Usually we are using sequencing, or we're using RT-PCR or conventional PCR for the gene expression. And here we got some pro-inflammatory cytokines that might link to the microRNA, and this microRNA can be important, like microRNA 15, 21, our intention to use it in the experimental setting. This was more like a prediction. Okay, and a little bit about bioinformatics, chemoinformatics, and biomedical informatics. A couple of words about the software design and the projects we have in my laboratory at ITMU University. Here is also the project relating to the blood-brain barrier here, and this is purely computational, and we try to implement the click reaction. The click reaction is a well-known reaction. Professor Bertozzi, Italian, she's Italian actually. She got the Nobel Prize for this particular reaction in 2010, and we are using it. And the idea is to generate a kind of a synthetic database and use this synthetic database to come up with the substances which are permeating to the blood-brain barrier. And my brightest students working on it 24/7, they just came up to the software. Of course, we have a backend. We've published backend, and this is the front end for overview for the doctors, for general practitioners, for scientists who is not familiar with the programming. And what is all about? We call it NeuroClick, because you're doing the click reaction. This is azide alkyne cycle addition. You are forming the cycle here, and this is it creates different isomers. One is 1.4 and the other is 1.5 isomer, and you have also cycle heterocycle, and these molecules might be promising. And what exactly we are doing? We are calculating some parameters like blood-brain barrier. This is the partitioning coefficient. Usually this is just this is very simple. You got the concentration in the blood and in the brain. Then you have the ratio, and then you do the logarithmic exchange, and you just come up with the blood log BB. It's called the log BB parameter. And this log BB can be computed if you know some descriptors of the molecules. For instance, if you know the lipophilicity of the molecule, and the lipophilicity is present by the log P, the partitioning coefficient, and you might use the second one, which is the polar surface area. And there are some models. These are regression models. They were developed in the past. They were the Richardson and Clark models. I was first using these models like 15 years ago in my first paper, where this was a kind of golden standard. Of course, now we have AI-based algorithms. Now I'm going to talk about it later, but these are also quite okay for testing, for the preliminary testing. And so you can see we are calculating different parameters, and everything is based on this particular coefficient, how to come up to the best molecules to go into the blood-brain barrier, for instance, to treat some cancers or neurodegenerative diseases like Alzheimer, for instance. And this is the interface. Everything is quite fast. So for example, if you want to generate like tens of thousands of molecules, it might take less than a minute. So it's relatively fast. Of course, we are going to parallelize it, and it will be even much faster than you can do for millions or hundreds of millions of molecules. And there will be the second. Right now, my student, my PhD student is working on it, and we are trying to integrate different modules. It's more sophisticated, so we can integrate the Ahmed paradigm with absorption. This